Discovery of DNA. What is life? Erwin Schrödinger, a Nobel Prize-winning physicist for his work on quantum mechanics, delivered a series of lectures in 1943 on how physics and chemistry describe life's processes. Life is subject to the same rules that inanimate objects are. He proposed that a molecule, an aperiodic crystal, contains genetic information due to the arrangement of its covalent bonds. An aperiodic crystal, unlike a crystal like salt that repeats the same structure, doesn't repeat its structure. Thus, it can code an infinite amount of information with just a few components. This is a good description of DNA, which is a giant molecule made up of only 4 different building blocks, nucleic acids. James Watson and Francis Crick, more on them later, wrote that they used his work as inspiration in coming up with the structure of DNA. After Griffith's experiment, most scientists believed that the chemical transforming bacteria was a protein, not a nucleic acid. Avery, McLeod, and McCarty used a biochemical fractionation approach to Griffith's experiment to isolate what they called the transforming principle. They wanted to determine whether the molecule responsible for virulence was lipid, carbohydrate, protein, RNA, or DNA. In 1944, they reported, based on numerous experiments, that DNA, or nucleic acids, was the cause of the transformation. Avery's team used a test tube assay. This is when a scientist compares differences in test tubes after treating each differently. The benefit is that more specific reactions can be found. This approach led to more information than dead or living mice can provide. First, the S strain or deadly bacteria were heat-killed and mixed with detergent. This caused the bacterial cells to break apart. Their membranes lysed and spilled out the cell's contents. The lysate was centrifuged at high speed. Centrifugation separates the lysate into a liquid supernatant and a solid precipitate, as you see in the image. The fluid in the upper portion of the test tube, the supernatant, contains less dense materials like proteins, enzymes, and nucleic acids. The precipitate contained the large organelles present in the cell. So Avery isolated the supernatant, removing the precipitate, to use because it contained smaller molecules like lipids, carbohydrates, RNA, proteins, and DNA that were more likely to be the genetic material. The supernatant was subjected to a series of biochemical treatments to eliminate one class of biomolecule at a time, and then treated for virulence. So first we remove lipids. So if we remove lipids, as each biomolecule was removed, like the lipids, the supernatant was mixed with a living R strain. If the supernatant still killed the mice, as would be the case in the lipids, that meant that the removed molecule was not involved in the R strain picking up the genetic material from the killed S strain. When the DNA was destroyed by DNA depolymerases, the mice lived. Without DNA from the S strain, the R strain did not become virulent. DNA is the transforming principle. It carries the genetic material. The definitive proof. The Hershey-Chase experiments were a series of experiments conducted in 1952 by Alfred Hershey and Martha Chase that confirmed DNA was the genetic material. By this time, many new discoveries allowed these scientists to go beyond what others had been able to discover about nucleic acids. Most importantly, intense research on viruses at the time expanded the knowledge of these tiny particles. Hershey and Chase concentrated on bacteriophages. Bacteriophages are small viruses that infect only bacteria. They are made of proteins and DNA. The bacteriophage inserts the DNA like a syringe so that only DNA goes into the bacteria and the protein structure stays outside. Scientists were very familiar with using radioactive techniques to observe particles that could not be seen. Viruses were too small to be seen, so Hershey and Chase used a novel approach that took advantage of radioactive labeling. This allowed them to track different parts of the virus by looking for radiation. They began by creating two kinds of a radioactive virus using a labeling technique. Below is how they made virus A. Using bacteriophages, bacteria, and radioactive sulfur in a petri dish, viruses grow via a lytic cycle. They need sulfur to make proteins. Radioactive sulfur was provided for the cycle to ensure for the radioactive labeling. Since it is the proteins and not the DNA that need sulfur to be constructed, only the proteins in these new phages are radioactive. So only the outer protein parts would show the radioactive marker. The procedure is repeated to make virus B, replacing radioactive sulfur with radioactive phosphorus. So the second type of virus was using radioactive phosphorus. They need phosphorus to make DNA. So radioactive phosphorus was provided for the cycle. Since DNA, not proteins, need phosphorus to be constructed, only the DNA in these new phages are radioactive. So in virus A, the protein needed the sulfur, so it would be radioactive. And in virus B, the DNA is what needed the phosphorus and would therefore be radioactive. Virus A was mixed with bacteria, infecting the bacteria with the bacteriophage, which then replicated inside the virus. The infected bacteria were then isolated and analyzed. They were not radioactive, indicating that there was no sulfur in the bacterial cells. That meant that the proteins did not communicate instructions on how to reproduce. That could mean that DNA was the vehicle for reproduction, but that had to be shown definitively, so it was necessary to use virus B. Virus B was then mixed with bacteria and absorbed by the bacteria, allowing it to become infected. The infected bacteria were then isolated from the virus and then analyzed. The remaining bacteria were radioactive, indicating that there was phosphorus in the bacterial cells. That phosphorus was attached to or labeling the DNA. That meant that the DNA was the vehicle for reproduction. The two trials definitively showed that DNA was the vehicle for reproduction. All right, here's a summary of the Hershey-Chase experiments. And there's an image here that we'll talk about, but it's larger on the next slide, so we'll look at it in more detail there. So the sulfur-labeled bacteriophages infected the cells and changed their genetic programming, even though no protein had entered them. And the phosphorus-labeled bacteriophages infected the cells, changed their programming, and made them radioactive, indicating that it was the DNA that had entered the cells. So here's virus A, where in red the sulfur is labeled, and this is the protein outer shell of this bacteriophage. It's allowed to infect the bacteria, and then we, through blending and centrifugation, we isolate the bacteria from the bacteriophage. After centrifugation, there's no sulfur in the cells, but the cells are able to replicate and become infected. Okay, so something is infecting the cells, but it is not a result of the protein capsule in red because there's no sulfur in the cells after we isolate them. In green, in virus B, the DNA is phosphorus labeled. Exact same process is followed of infection, blending, and centrifugation. And once we isolate the bacteriophage from the bacteria, there is phosphorus in the cells, indicating the presence of the DNA from the bacteriophage. So the DNA is what is entering the cells and is the vehicle for reproduction. Chargaff's rules. With the data collected by Hershey and Chase, the focus of the scientific community shifted to nucleic acids. DNA in particular became the focus of scientists looking to make the next big discovery. In the late 1940s, Erwin Chargaff discovered that the amount of guanine approximately equals the amount of cytosine. For humans, the percentages are 19.9% to 19.8%. And the amount of adenine approximately equals the amount of thiamine in a species. For humans, 30.9% adenine, 29.4% thiamine. This information would be used by James Watson and Francis Crick in determining the structure of DNA. Working at King's College London in 1951, Rosalind Franklin and Maurice Wilkins produced X-ray diffraction images of DNA to try to discover its shape and understand its mechanisms. The DNA image above, photo 51, was created by Franklin and was the clearest picture of DNA, allowing Watson and Crick to propose its double helix structure. X-ray diffraction shoots subatomic particles into a substance. The collision of these particles with those of the molecules in the substance cause them to diffract or ricochet at specific angles. This gives insight into the structure of the substance. So here's the X-ray being shot at the substance, resulting in these specific angles of diffraction. And there's a paper here that's sensitive to the X-ray. Just think of like X-ray papers that you've seen like at a doctor's office. But that results, if I take this photo paper and just make it flat so I can see it, this is the result that I see in photo 51. The electron micrograph of fruit fly DNA shows several replication bubbles, indicated by these yellow arrows. You can see the bubble here, 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 and here, along with the replication forks, so you can see them at either end, where the DNA is unwound. Eukaryotic DNA has multiple replication forks. Prokaryotic DNA have only one replication fork. Watson and Crick deciphered from photo 51 that DNA was a double helix. They began to build models of the structure so they could speculate on how DNA can both self-replicate and contain the code for all the traits of living things. Topoisomerase, which you see indicated by this yellow bar here, is an enzyme whose purpose is to uncoil the DNA at that point so that each strand can undergo replication. Once unwound, helicase breaks the weak hydrogen bonds that hold nucleotides together, opening up both strands to become templates for new strands. Helicase is the molecule that creates the replication fork. So you see that helicase right here.